So we can get started. Today's oral lecture. And the first lecture is the plenary lecture by Professor Aleksandra Walczak from Paris, from École Normale Supérieure. And the title of her talk is Prediction in Immune Repertoire. Please, Aleksandra. We are ready. Hello? Hello?
We can hear you, but Aleksandra is probably not listening.
Aleksandra is not. You can. Okay.
Chat.
Yes, Aleksandra, we are ready. You can get started, please.
Hi, sorry, I couldn't hear you before.
No, we are ready. You can get started. Go ahead.
Okay, great. So sorry about that. Thank you. Okay, so hi, my name is Aleksandra Walczak. I'm really happy to be talking to you today. So I'm going to tell you a little bit about immune repertoires and hopefully try to convince you it's a complex system. Although I usually try to convince audiences quite the opposite, that it's not as complex a system as they initially thought. So this is work that I've been doing over many years now with Thierry Morat, who's also at DNS. And the first part of the work that I'll talk about is with our collaborators in Moscow. It's work led by Anastasia Minervina and Mikhail Pogorelli, with Maryam Bensouda, who's at DNS also. And then I'll talk about some more theoretical work with Andreas Meyer and Vijay Palasar. Okay, so I'll start with talking about COVID, because I guess everybody is somewhat interested about that these days. This will be a very different point of view than you've been hearing about probably at this conference. So no epidemiology, we're going to get more into each individual. So we have two people who had the good idea to go skiing last February, and then they came back with something more than expected, were put into quarantine, and at the end of quarantine donated blood samples at different time post-infection. And from these blood samples, we pulled out the T cells, so cells that interact directly with pathogens and recognize them and sequence their receptors. Okay, so T cells have receptors that are diverse, and these diverse receptors protect you against various pathogens. That's what we call a repertoire, and it's the diversity that protects you against the different pathogens. And by looking at the diversity of the different receptors, so the list and their abundances, so that's what you see here, which we can get out of sequencing of these cells, we can figure out which cells and which sets of cells are needed for protection. So if you look at the abundances of these different receptors, of these different cells at different days after the infection, this is what the data looks like. Okay, so I'm showing this to you to say it looks like a mess and you can't figure anything out. From here, what you would hope to see is something shooting up, which says this is the receptor. Open window, bottom right, blocking the slideshow. Okay, I don't see it. So I don't really know what to do about that. Let me stop sharing and start sharing again. Let me just share the whole desktop. Right. Is this better?
Yes, much better.
Great. Okay.
We can see now, yes.
Okay, great. Sorry. So anyway, two people, data points of the T cell receptor sequences after the infection, and what you hope to see. So basically you have 10 to the 9 different cells, 10 to the 11 total, 10 to the 9 different receptors, 10 to the 11 different total receptors. You sequence a sample, a large sample of the order of 10 to the 6, 10 to the 5, 10 to the 6, and you hope to see a peak, well, a peak more that looks like this, because that's the response to COVID. Now, so if you look at the data in MS, so the first thing we did is we did a first try of just doing PCA on the trajectories, principal component analysis, and you see clusters emerge. So you see a cluster, so this is time, PCA on time dependent on the whole trajectories, and you see one cluster which basically doesn't have any information, doesn't seem to change, so there's no response. You see the kind of cluster that corresponds to the response you expect, that there was a peak at day 15 and then decayed. And then you see a second peak, which you didn't, which nobody really expected at day 37. Okay, so we do see some signals. So basically from the mess, we're able to extract some information, but then we wanted to analyze it in more detail. So these are the kinds of trends we're going to be looking at. So the first thing to realize about this data, so this is just a sort of a cartoon showing that there's many cells. Each cell has one type of receptor, but there are many types of cells with many types of receptor. This is what we're trying to get at. This is what we get in as data, is reads of the mRNA that encodes for these sequences. And the first thing to realize is, again, I already made this point that the data is noisy. So even if we look at the same person, who I call Terry here, at the same day, day 15, same time, we look at 2 different replicates from this person, and we look at the counts in one sample and the other, in one replicate versus the other, we see that it's very noisy. Basically, all this data should be falling on the diagonal because you're just taking 2 technical replicates. But you see that's not true. There's some clones that are in one sample have 0 and in the other have 100 counts. So that gives you an idea of the noise of the system, and the noise is twofold. There's of course experimental noise, which comes from sampling, which we'll model in terms of Poisson sampling, because the first thing we're trying to do is calibrate this experimental noise and build a noise model. So it comes from sampling, but the second type of noise is biological noise, which comes from the fact that these receptors are encoded in DNA, and then which is to make the protein, you need to make mRNA, but mRNA is differentially expressed in different cells. So you get intermittent expression with different burst sizes. So we'll model that as a negative binomial kind of noise. And to make things even more complicated, the original distribution of these clone sizes, so how many times you see a given receptor, is a power law. So basically what we do when we model the noise is we say that our data is really coming from an initial power law of frequencies, from which we sample differentially frequencies due to the mRNA production, and we sample and then they're sampled on top of that differentially due to noise. Once we build this noise model, we're now ready to do a probabilistic inference of the response. So to do that, we model expansion simply as exponential expansion. And we say that some part of the cells will expand and some part of the cells will not expand. And then we calculate the a posteriori probability of expanded clones. And we say that, well, they're expanded above some significance level. And so we can get a list of expanded clones. And again, we can plot them as a function of time. And so this is day 0 now and day 15. So before I was just showing you data from day 15 because that's what we did the PCA on. But, and you see these are decaying clones, but you also see, we also have data luckily for these people at a year and 2 years before the infection. So we can actually verify that these are not just clones that were very large for a very long time, but they went down. There's another question. Okay, it's the same thing. And we see the same thing for 2 people. Okay, so we see that there's this expansion, this expansion that happens at day 15 and then goes back again. So we're able to identify a large expansion. And if we look in more detail at this large expansion, we see that some of these cells have what are called the memory phenotype. So, and this is something that you can say from the experiment in other ways, but basically this means that they'll hang around for a long time. And memory cells, and specifically one type that we identify here, are cells that confer immunity, and they give you stable immunity post-infection. So we see that these people who, I forgot to tell you, suffered a mild COVID infection actually will have stable post-infection immunity. We don't know how stable, but from the order of time we looked at, it's stable. And the important statement here is that, you probably hear a lot in the news about antibody immunity. So antibodies that come from B cells, these are T cells. So this tells you that there's another way your body can produce immunity against COVID. But the most interesting thing here is that if we look at these time points before infection, these people already had immunity against COVID. So maybe that's why they suffered a mild infection, and we'll get back to that. So I described the general theoretical framework for inferring what's a potentially responding receptor sequence, but we don't actually, this is a theoretical framework. We would like to validate it and say whether these sequences are really COVID specific, because all I showed you is that they're responding, they're changing with time. So luckily, Pat, one of our donors, participated in another study 20 days after their infection. And in this study, they did a sort of, there was an experimental study where they did a sort of fishing exhibition, meaning they put a COVID epitope or a COVID protein onto a kind of molecular fishing rod, and they stuck it into a sample taken from Pat's blood. And then they saw what stuck to the COVID protein. So basically which T cells recognize this COVID protein. So this is the specific COVID protein they tried. And as you see, they found cells that were responding. Now we looked at that list of cells and matched them across the cells that we identified as responding in our, at all the cells that we measured in Pat's system. And this reproduces this blue curve, which is similar to the curve we saw before, okay? So this is not all the data because they're only testing one COVID protein, whereas Pat's overall response was probably to a few proteins, but it reproduces the trend. And good news for our theoretical method, it actually, all the points in green are the ones that we identified independently just using our theoretical method. And we're managing to basically catch all the high frequency receptors here. So our theoretical method, although it does not use any molecular information, is still able to identify a response, and it's able to do it in a patient-dependent manner. Okay, so that's the validation step. Now let's get into a little bit into interpretation and another kind of validation that maybe will appeal a little bit more to you since it's network-based. And so here we turned to a database that was recently published of T cells that respond to different COVID antigens. So they did like a large-scale fishing expedition where they, so this is adaptive technologies where they took many COVID proteins and tested them against many people's blood and saw what responded. So at the end of the day, what they get is a list of responding T cells combined with the actual COVID proteins, epitopes that they respond to. And so we combined the responding T cells from this large-scale fishing exhibition mirror with the responding T cells that we found in our study and built similarity networks. Means we put a link between all T cells that differed by at most 2 amino acids, and this way we build these similarity clusters. And thanks to that, we were able to identify, we found many clusters, but for 32 of these clusters, we could link them. So the clusters are clusters of T cells, but then thanks to this study, we could link them to specific COVID epitopes or proteins that they respond to. So now we know what they're specific for. So this is what our data look. This is an example of a certain subset in PAD again. And we see, so this is an example of a cluster, and these T cells here respond specifically to this COVID epitope. Now it turns out that the response of the T cells in this cluster make up 21% of all of PAD's.
Good morning, puppy.
Okay, of all of PAD's response. So it's a huge, this epitope is driving a huge part of his response. This is the logo for the T cell that's responding. But what's interesting about this is that the T cells that respond to this epitope, so the T cells here, were already in PAD's repertoire and PAD's blood a year before the COVID infection. And it turns out that if you mutate only one of the amino acids in this protein, you get a common cold protein. So maybe PAD had a common cold a year or before the infection, and maybe this is what drove then the mild infection. So you can see through network analysis, we can try to get these kinds of things, but what we can also do is we can do diagnostics. So the question we specifically ask is, since we can find these different motifs, T cell motifs in PAD and TERI, can we now identify, if we look at the blood of many people, can we just identify who had COVID and who didn't have COVID based on the motifs we found? So we took one of TERI's motifs, and we asked whether this is true, whether we just by looking at the different people in yet another public database that now is here, Nolan et al., can we figure out who had COVID and who didn't have COVID? So this is a database of people who had COVID, and this is a control database, Emerson et al., from the same group of people who didn't have COVID, and we ask whether we can figure out the true positive versus the false positive, and the answer is no. So then we realized we thought, well, what if we restricted to people who have the same presentation machinery? So what you should know is that for T cells, the epitopes or the proteins, the COVID proteins or any other proteins get presented on the machinery which is called HLA. HLA is basically the way, the way you're probably familiar with it is if you're getting a transplant, you need to get your donor to match, and this is what your donor needs to match. It needs to, among other things, it needs to match your HLA type, but this is basically a machinery on cells that presents proteins. Now, so we thought if we restricted to people that had a similar makeup or the same makeup as TERI for whose motifs we're trying out, then maybe we'd do better. Now, the problem is in this data set of the COVID donors, we didn't have information about what the scaffolding, what this HLA type is. So we used an HLA inference method where we basically matched people's T cells. Now, forgetting about COVID, forgetting anything, we just take a database where we know, so actually in Emerson, we know what the HLA type is, and we just take one person's T cells and match it against other people, and this way we infer what the HLA makeup in this data set is. And so that works, and it gives us hope, because if we now compare people with COVID to control donors and ask how many of them have a given motif, if we restrict to people who have the same HLA type, things are starting to, there is a difference between patients and not, COVID patients and not. And if we now do our true positive versus false positive curve, we actually do see signal. So basically, if we restrict to people who have the same HLA type, we're able, using these motifs, just to say whether somebody had COVID or not. So that's sort of encouraging, and it's the same thing for PAD. So as you can see, this motif thing can actually be used. Okay, so let me now change gear a little bit and tell you about another type of line of approach that we've been doing. So where we basically ask, do people who have a given disease, well, or a given infection, for example, here, COVID, do they share more sequences, more T-cell sequences or more B-cell sequences than expected by chance? And to answer that question, we first need to figure out how many sequences are you going to share by chance? And so how are we going to do that? Well, to do that, we first have to answer the question of what's the probability that you're going to have any T-cell sequence to begin with. And so that's the first thing we did. We took data from healthy donors and we tried to answer the question, what's the probability of you having and producing and keeping a specific T-cell sequence? So what is a T-cell receptor sequence? Well, a T-cell receptor sequence is made out of 3 parts. They're called V, D, and J. And between them, so these 3 parts come from genomic templates. So as I told you, there are 10 to the 9 different receptors you have. You couldn't encode this directly by genes. That's too many genes. So what happens is you have an editing process where you have a certain number of templates for each of these. And then you edit the DNA, you cut the DNA and just pick 1 V, 1 D, 1 J. And then there's additional insertions and deletions that come in. So just the combinatorics takes you to the order of 1,000 to get to the 10 to the 9th, that's 6 orders of magnitude extra. Those are the stochastic non-templated insertions and deletions. And some of these rearrangements produce what are called out-of-frame reads, means reads that will never form a protein. And we use those reads because since they'll never form a protein, nothing else will ever happen to them. So that's information simply about the generation process. And by nothing else, I mean selection. So we use these out-of-frame reads to figure out what's the probability of generating each one of these receptor sequences. So for a given for each sequence, we build a library of possible scenarios, meaning V, D, and A choices and number of insertions and deletions that's consistent with this sequence. And we assign weights to it. And we do it for all sequences, and then we calculate marginal observables, such as the probability of using a V gene, probability of having a certain number of insertions at this junction, deletions, and so on. And then we have a model, which is the most general factorializable model that is consistent with the known biology to recalculate these weights. And we do this self-consistently until convergence. Once we have it, we know the weights, we can sum them up and say what's the probability of generating a given sequence. So that's the general idea. So in short, we have a way for figuring out from the data with a model what's the probability of generating any given sequence. So just to give you an idea what this looks like, this is the probability of insertion. So the number of insertions and how many times you see a given number of insertions. And you see that the error bars here for the blue are the VD and the red are the DJ are over 9 real people. So the process is very universal, very reproducible. The dashed lines here just show what would happen if you didn't sum over all these other sequences, if you didn't do a Bayesian approach. You see you get this spurious peak, so you really have to do this. And if you then look at the histogram or the density of the probability of generation of a given sequence. So you ask how many sequences in your body do you have that have a generation probability that's 10 to the minus 10, 10 to the minus 12, 10 to the minus 15, and so on. So you make a distribution like that. You see that it goes over 20 orders of magnitude. So some sequences are much more likely to be generated by any of us, since this is a universal distribution, than others. But overall, we now have a way of saying what's the probability that you or I generate any T-cell receptor sequence. And we also have a probability to say that I won't describe how we did it, but we'll, now using the actual data, the receptors that make proteins, so the in-frame data, we can say what's the probability with which they are selected. And at the end of the day, I can now tell you what's the probability that you see a given receptor, which is just the probability with which it was generated times the probability of selection. So we can do this, and we can ask, so if I know that, what's the probability that you have a given receptor and I have a given receptor, then I can calculate the probability that we share a given receptor just by chance. So that's just the square of this P receptor, because our two processes are independent. So from that, we can also calculate how many sequences we actually share. So, so this is the number of sequences shared as a function of how many people share them. So this is shared by 2 people, 3 people, up to of the order of 700 people. And you see that basically we can completely predict how many sequences are shared between, you know, of the order of 1,000 people in the world. And the same thing is true for T cells and B cells. This is human data. But if we look at B cells from people that had COVID, we see that we underestimate the number of shared sequences compared to what we see in the data. So it seems people that do get COVID infections do seem to generate very similar responses. And this is consistent with other data, that if people look at these B cells, they're not very much mutated. So B cells, when they respond, they usually mutate, but these B cells from people who had COVID are not very mutated. So that means it's a very sort of basic response. Okay, so where am I? Chat. Okay. So now I'll shift gears and I'll tell you about the completely different way of thinking about the immune system, but I hope also as a complex system. And where we ask the question whether the immune system can be thought of as an optimal predictor of future pathogens. So meaning if we have a given set of infections now and we have an immune system that works against it, so we have this diversity of different receptors that protects us, how should the abundances of these different receptors and their identity change as a function of time to protect us against future infections? Now, why is this even something to think about? Well, COVID may actually ruin the intuition here, but if you think about something like flu, pathogens evolve in time. And so since they evolve in time, they evolve according to stochastic processes that are correlated in time. So the basic idea is, can we exploit the correlation in the evolution of the pathogens to predict what would be the optimal thing for the evolution of the immune system to do? Okay, because they can also evolve, these cells can proliferate, these cells can die, and new cells can be generated. So how should this work? Okay, so the name of the game really that we're trying to play is, can we build an immune system that is able to estimate the future frequency of pathogens and be prepared for it, right? Because that's really what is optimal here. So the way we're going to set up this problem is we're going to say we have a receptor distribution P of T, and we have a distribution of pathogen frequencies Q of T. And we know that pathogen frequencies evolve according to stochastic population dynamics. So there's some operator A, which you can think about as sort of Wright-Fisher dynamics, or just mutations and then stochastic growth. Well, I actually just mainly increase, stochastic growth, let's say. But let's say we know this evolutionary dynamics. The question now is, how should the optimal repertoire respond? And so what does optimal here mean? Well, we're going to say that it means that the expected cost of an infection is minimized. So what's the repertoire that minimizes the cost of infection? Of course, the problem is that we don't know what is the actual, what is Q of T, what are the pathogen frequencies at the given time. So we're going to say that we can estimate them. So we're going to put in a belief function for Q. So a meta probability distribution, Q is already probability distribution. We're going to introduce a distribution over distributions that is going to average over all possible future distributions of the pathogenic environment. And so we're going to basically say that the immune system keeps this belief distribution of what the state of the pathogenic environment could be, that it propagates also in time according to the same operator, to the same dynamics as the actual pathogen evolution. And then we're going to say that when an infection happens, what an infection is an actual collapse of the distribution. So now for a given Q, so for a given pathogen, we're going to say that instead of having a probability of seeing this pathogen, you actually know whether you saw it or you didn't see it. So you collapse the function. And so all in all, we're going to say that propagating, that prediction is propagating the belief in time, which is a mixture of these propagating the prior and the encounters. And the reason the immune system can do it is because it has this thing I already mentioned, the memory repertoire. It has a long-lived subset of cells that know what they've seen before. Okay, so how do we define this cost? Well, if we take now a pathogen distribution at any given time, we're going to say that pathogens and receptors live in the same space. That you can think in simplicity about the two-dimensional space, and the closer they are together in this space, the more likely they are to recognize each other. So this is sort of depicted here, that if I have what's it called? Blue receptor here, then it can recognize all pathogens within this blue bubble, and same thing for the red one. But there's a concept in immunology of cross-reactivity, meaning that these two receptors, of course, they're best suited for things that are closest to them, but they'll also be able to recognize things in between. So any pathogen in between here in this area will be recognized by both the red and the blue, just with a lower probability. So based on that, we can calculate the probability of an immune response from an encounter with a given pathogen by summing over all the receptors modulo these clouds. So you can think about them as Gaussian clouds. They don't have to be Gaussian over all the receptors. So that gives you the probability of recognizing a given antigen, which is a given pathogen, so one bug. Then we're going to measure encounters in time and the number of encounters, and we're going to encode the fact that the cost of infection generally doesn't decrease as a function of time. So you see a pathogen, you try to find a receptor that'll recognize it, and as you don't, you're generally not getting any better until you recognize it. So we encode this by saying there's a Poisson distribution of recognition events with this probability, and then there's some infection cost that is a non-decreasing function of time, and we integrate over all times until recognition. And then we can correct that maybe some bugs are more important to recognize than others through a virulence factor. So this is for one pathogen. Now we have to sum over all pathogens to get the cost, and then we minimize it. So when we do that, we can start answering questions. And the first question we can ask is, does memory help? And it turns out it helps a lot in sparse environments. So if we look at how the cost decreases from not having any memory, so not doing any of this prediction, not keeping the belief about the pathogen distribution, we can see that it goes down as a function of basically of sampling non-negligible pathogens, but it goes down. So it goes down both as a function of pathogen sparseness and as a function of sampling. So the more you sample, the smaller your cost is, which makes sense. But it's also much easier to recognize. The cost goes down much faster in sparse environments, and so basically you can do, there's different parameters in this problem that I don't want to go into in detail now, but there's the how fast does the environment change, how how sparse, meaning, again, think about the two-dimensional space. So how much is the space covered, how many pathogens there are, but everything collapses on one curve, and the only two parameters that matter are pathogen sparseness and sampling. And so this is reminiscent of compressive sensing, where you're trying to encode a lot of, well, reconstruct information, and it's actually much easier to do if the problem is sparse. And so we think that this may be the idea of how can you learn to protect yourself in a situation where you have many pathogens. Okay, I'm done. If the thing is basically, you can do it because the system is sparse. And so we also think that, so if you look at how this function happens in age, you first learn the features of a sparse environment, and that's a fast learning where your memory fraction goes down and your susceptibility to pathogen goes down very quickly, and then it levels off. And if you ask how well, maybe I'll skip this. So you can also ask, how do these distributions actually look like? So I told you to think about the two-dimensional space, but here I'm plotting a one-dimensional space. So the antigen distribution is in blue and optimal repertoire is in green. And you see the optimal repertoire is peak. So you place your pathogens at discrete time points, and that gives you, nevertheless, very good coverage against different pathogens. So you can ask the question whether two people living in the same environment should see the same distribution or different distribution of pathogens. And the answer is should have the same, that see the slightly different distributions of pathogens should have the same optimal immune system or different immune systems. And the answer is they can have very different immune systems. So the similarity, the overlap measure between the immune systems is very different, although they essentially see the same environment and have practically the same coverage. And the reason is this peak distribution and this cross-reactivity. It doesn't really matter where you put your receptor, because you will be protected due to these little Gaussian clouds of cross-reactivity. So whether you have a repertoire with receptors placed like this or like that, from afar it looks the same, it covers space, which means there are many different solutions to the same problem. And if we look at back at the question of how many receptors are shared between two people, as I said, we have these random expectations and we see the observation, they match very well, but the numbers are small. You see, of course, they depend on sampling, but nevertheless, for sampling of tens of thousands of receptors, we only share a few hundred. So we have this small but predictable overlap because we can all find very different solutions to the same problem. So thank you. I think I'm out of time. Sorry for the technical problems.
Finished?
Yes.
Okay. Thank you very much, Aleksandra. Perfect timing. Couldn't be any better. The talk is now open for questions. I do not see, here is a question. Let me phrase it for you. Could there also be some advantage in different people having different repertoire, for example, for group immunity?
Different people. Yeah, no, definitely. Okay, so where do I start with this? With this question. I mean, I think what I'm trying to say is in a way by construction, we have different repertoires. Now, what you're saying is, can there be, is it good? I mean, there's the simple advantage that, if we have different repertoires and my repertoire fails to combat the bug, if we had exactly the same repertoires, this would mean that you're doomed too. And if you have a different repertoire, you don't have to be doomed. So I think that's the obvious advantage for group immunity. I mean, there's also a disadvantage that, I mean, okay, if you mean group immunity in the sense of that you know, in the epidemiological sense, then in a way it doesn't matter where your immunity comes from, as long as the virus does not manage to spread. So the danger of somebody having a repertoire that is still susceptible is basically that it opens up a susceptible pool that would otherwise not be there. But I think that this is a very deep question in a way, because there's two levels of the repertoire. There's the molecular level of actually encoding in the receptor, and there's the phenotypic level of actually giving you immunity. So one statement is that, so the second part of the talk was at the phenotypic level. So even at the phenotypic level, we can have very different types of solutions that nevertheless give us, if they give us immunity against the same pathogen, then you know, that from the level of sort of epidemiology, it doesn't matter whether it's the same or it's different. So in a way, there are three levels.
Okay. Any more points, comments, questions? Feel free to unmute yourself. If no, then we will proceed to a short break. We will meet again in the parallel rooms, the 6 parallel rooms, in about 15 minutes from now. Thank you very much, Aleksandra. Thank you very much for everything.
Thank you.
I have a question.
Oh, you have a question? Okay.
Oh, I'll just, can I still?
Yes, yes, of course. Go ahead.
Is there any implications of the logic of the study for microbial resistance? And
you could, okay. Yeah, so let me just quickly say, you could use the theoretical framework that we introduced. You could, and people are thinking in that way also about microbial resistance. And can you hear me?
Okay. Well, I'll leave the room open anyway, because this room will be parallel room number 1. So this room will stay open, whoever wants to stay, but the break is right now on. So feel free to go and fetch some coffee for you. See you in a few minutes.
Sorry, did you have a second part to your question? Okay.
Well, if Mauricio is gone, I might have a question.
All right.
What would be the take-home message for like more general public from the research about?
Well, I think that, you know, one take-home message is that you can have very different, first of all, phenotypic solutions, but then definitely molecular solutions. I mean, I didn't really have time to sort of paint the expectations of what the general public feels right now, but the viewpoint of not just the general public, but also many doctors, is that is a sort of lock and key mechanism, and there's a golden bullet receptor that protects you against this bug, right? And of course, if you're designing a vaccine, well, you know, you kind of have to have that framework because you have to produce a molecule that'll go into the vaccine. But I think the big picture message that we're propagating is that this is really a very dynamical system. It's a self-organized system. So it doesn't have to work on golden bullet levels. That you could have a vaccine that doesn't squish the bug, but it activates an existing system to stimulate it to protect yourself, and the protection that will stimulate in you and me will be different. In a way, this is also what vaccines do, but you could imagine, you know, because what vaccines do is they stimulate something that's already in you, but I mean, at least some vaccines. But you could imagine, you know, really taking this further and I mean, in a way, this is what immunotherapies are trying to do, to stimulate solutions, basically to enable the body to find solutions rather than give it solutions. Is that big picture enough?
Thanks a lot.
I also have a
Hi.
question, if I may. So I was thinking about this, if you follow like the grand stories like Jared Diamond's Guns, Germs, and Steel, where supposedly in the Americas, they were less resistant against the pathogens coming in. If one could surmise, like listening to your talk, that there is obviously a coevolution between the pathogens and the immune system going on. That is not like sort of like purely genetic, in a sense, like bound to our own cells, but there may be something like, is there some way to give a population that didn't have the 20,000 years to sort of like have the same immune cells, to give that population the right kickstart, to have the right receptors to then co-evolve with whatever the current flu does or something?
Yeah, so no, this is a great question that I think we could spend hours on. I mean, again, the short answer is vaccines.
Okay.
Right? I mean, I think this is what we do to our kids. Mm-hmm. So, I mean, okay, where do I start? The first thing is that the immune system is somatic, so you don't pass it on to your kids. Okay? So in a way, however, as you point out, though the European populations were immune against smallpox, because as a population we've evolved with them. So there's an interesting, I mean, this is why this is such a big question. There's a level of like what actually evolves on real evolutionary timescales, and that's the machinery, right? This VDJ choice machinery. If you look at mouse have the same kind of machinery, but it's slightly different than ours. It's evolved. So I think that's like one really super interesting topic of research, of how does the environment shape this machinery on long time scales. Then if we look into us, we do have somatic evolution. And this is another huge area of research of aging of the immune system, because each one of us is a walking evolutionary experiment of co-evolution going on between the pathogens that we encounter and the immune cells. So if you look at yourself at 80 versus yourself at 5, what has changed, right? Unfortunately, nobody has lived long enough yet to have done this experiment. But that's the sort of ideal experiment, and we are trying to do that more locally. We're looking at how the immune system of people changes over a few years, exactly what changes, how does it depend on the age of the person. We know various things. But yeah, on the other hand, there is this co-evolutionary aspect, which I'm very interested in. And this is sort of what led me to also looking at COVID is, how do we co-evolve? And again, as a population. So for example, the flu evolves at the population level. Nothing much happens in you yourself, but it's all of us as a population that exert an immune pressure. So like the first question I got asked, right? Somebody's immune, so the flu cannot attack that person. Somebody else also, the flu's going to try and find the given flu strain, has a game of finding a person who is not immune to it so that it can survive. That's the game. But on the other hand, our immune systems have the game of, once we get it, to quench it as quickly as possible. And so that's the co-evolution thing. Now, if you ask, how do you give somebody this, how is it exactly that we transmit immunity long-term, and how did we do it before vaccines? I think the simple answer is we were not very efficient, although it's clear we did do it because of smallpox. Now, the other thing about smallpox to remember is that this is the adaptive immune system. Before the adaptive immune system sets in, there's something called an innate immune system. and that's much faster, and it's not, so that doesn't evolve within you, but that's probably where, you know, things like you know, I don't actually, honestly, I don't know where smallpox immunity comes from.
Okay.
But you know, I would guess that may have played a role too. But yeah, that's a really cool question. But if I had to protect the inhabitants of Papua New Guinea right now, for example, the people, you know, as you read Jared Diamond, so you know about the, you know, the people we haven't massacred yet. I think vaccines would be a safe bet.
And that could also be the massacre, the vaccine, right? So.
No, I mean, look, you know, as for the question for the general public, I would say, yeah, vaccinate your kids. That's
of course.
the message.
Yeah. Thank you very much. Consider my mind blown. And yes, it's like, it's one of the, you go to conferences not for the talks, but for the coffee break conversation.
Yeah, no, I know.
Conversation was totally worth $140. Thank you very much. I shall say, by the way. Thank you very much. Okay.